Welcome, everyone. Thank you for joining us in our second Young ISSAD Community Seminar Series. In today's seminar, we'll discuss Group B Streptococcus maternal vaccination. And we have as speakers Dr. Natalie Silmon de Monerri from Pfizer's GBS vaccine program and Dr. Simon Procter from London School of Hygiene and Tropical Medicine, joining us for our second event. Dr. Natalie Silmon de Monerri is the research scientific lead for the Group B Streptococcus vaccine program at Pfizer and molecular biology team lead supporting early and late stage vaccine programs. With over 15 years of scientific experience in academia and industry research and development. Dr. Simon Procter works in the Department of Infectious Disease Epidemiology at the London School of Hygiene and Tropical Medicine. And his research focuses on health economics, epidemiology, and mathematical modeling. So now I would like to invite Dr. Natalie Silmon de Monerri for her talk.
Good morning, everyone. Thank you for the kind introduction and the opportunity to talk about our work today. So I'm going to be giving an overview of progress towards developing a maternal vaccine to protect against invasive GBS disease in early infancy. So if we go to the next slide, please. The next slide, thanks. Thank you. So at Pfizer, we've been developing vaccine for over 170 years, beginning with development of the smallpox vaccine at the Lancaster County Vaccine Farm in 1882, followed by the diphtheria antitoxin, which was the first FDA licensed product in 1906. And Pfizer was the first company to commercialize a 7-valent pneumococcal conjugate vaccine to prevent invasive pneumococcal disease in infants and young children. After that, and if we fast forward to 2020, 2021, we and BioNTech gained approval of the first COVID-19 vaccine, as well as an extended valency pneumococcal vaccine for prevention against invasive pneumococcal disease in adults. Next slide, please. So we're harnessing this experience with vaccine development to expand our portfolio to look at maternal immunization. So vaccines against tetanus, influenza, and pertussis have been used in pregnant women for many, many years. And we are using that approach to develop new vaccines for Group B Streptococcus as well as respiratory syncytial virus. And how does this work? Well, maternal vaccines take advantage of the natural ability of pregnant women to protect their infants via transfer of antibodies selectively through the placenta to the infant. If you could hit the next button, please. Thank you. And so this process is upregulated during pregnancy, particularly in the second and third trimesters of pregnancy, where you see a significant rise in the IgG concentration in infants. And what this enables us to do with vaccination is boost those antibody levels so they're transferred to the infants to protect them during the period of risk of invasive disease, which tends to be in the first few months of life. And so with vaccination of the mom, we're looking to prevent the infant during that risk of susceptibility by boosting antibody levels. And this is the approach that we're taking for the GBS vaccine. Next slide, please. I think we need very little introduction for Group B Streptococcus for this audience, but just as a reminder, there are 10 serotypes that are associated with disease that are associated with disease, and the serotype distribution is globally is shown in the pie chart in the top there, with a predominance for serotype 3. 6 serotypes constitute 98% of invasive disease globally, and GBS is found to colonize vaginal and rectal tracts of approximately 1 in 4 pregnant women globally, and this is the major risk factor for development of invasive GBS disease in neonates. Disease occurs within 90 days of age and is characterized in two different forms. The early onset disease, which occurs between 0 and 6 days of life, or late onset disease, which is between 7 and 89 days of life. And importantly, infection in the neonate is associated with neurodevelopmental impairment, as well as stillbirth, maternal, preterm birth and maternal infections. And there's a significant GBS burden in low middle income as well as high income countries. And a major challenge for the field is that the standard of care varies globally. There is either risk-based or universal screening of pregnant women to detect colonization during pregnancy, and then followed by administration of antibiotics during labor. However, this standard of care varies significantly across the world, and even optimal implementation of intrapartum antibiotic prophylaxis approaches faces significant limitations. And so this is an example in the graph shown on the right of the case in the United States, the number of cases of GBS disease per 1,000 live births over the past 20 years. And so so we're looking at early onset disease in red and late onset disease in blue. And following the introduction of screening approaches and intrapartum antibiotic prophylaxis, we've seen a massive reduction in early onset disease, but really no impact on late onset disease. And importantly, for early onset disease, a burden still remains. And currently, there are no licensed vaccines to prevent against Group B Streptococcus disease. And for this reason, Pfizer is developing a 6-valent polysaccharide conjugate vaccine, which we call GBS6, to prevent against invasive GBS disease in neonates through maternal immunization. Next slide, please. So in the 1970s, Carol Baker published a seminal study showing that protection against neonatal disease is mediated by antipolysaccharide IgG. And so she looked at infants with GBS disease, and the proportion of the mothers that have anti-capsule polysaccharide antibody is shown here, where none of the infants that had GBS disease had anti-capsular polysaccharide antibodies, and the infants that did not develop disease had antibodies. And on this premise, several vaccine candidates have been developed for on the basis of anti-polysaccharide conjugate vaccines. Next slide, please. So we've been developing GBS6 for a number of years. In preclinical studies, we've shown proof of concept with GBS6 inducing robust binding and functional antibody responses as measured by opsonophagocytic assays in mice as well as non-human primates. When we've shown this by active immunization with GBS6 or passive transfer of anti-polysaccharide antibodies or human vaccine sera, and these protect against challenge in a mouse neonatal sepsis model, which is outlined here, wherein we immunize mice with GBS6, we mate the mice, and then take the pregnant, and then the mouse becomes pregnant. And then we challenge the pups with a high dose of each different serotype of GBS. And the survival plot is shown below. In comparison to PBS, we see very high rates of protection in this mouse neonatal sepsis model. Next slide, please. So in terms of our clinical development, we've completed a phase 1 first in human study in 365 adult males and non-pregnant women in the United States. And I'll show you some data on that in the next slide. We are currently undertaking a phase 1/2 proof of concept study in pregnant women and their infants. And this is ongoing in South Africa, the UK, and the United States. We've completed a phase 2 study looking to see whether we can boost antibody responses in adult males and non-pregnant women in the United States who were previously vaccinated in the first in human study. And we're also evaluating co-administration of GBS6 with Tdap vaccine. And this is ongoing. Next slide, please. So these are data from our first in human study in non-pregnant adults. For each of the different serotypes here, you're looking at the geometric mean concentration of anti-polysaccharide antibody concentrations over time. following immunization. This is a single-dose vaccine, looking at different formulations, 5, 10, and 20 microgram with and without aluminum phosphate compared to placebo. And as you can see, you rapidly after immunization see a robust rise in antibody response that's maintained throughout 6 months. And this study has been published in the Lancet Infectious Diseases in 2021 and supported further evaluation of GBS6 in pregnant women in our phase 1/2 study. Next slide, please. So the phase 1/2 study is being undertaken in South Africa, the UK, and the United States. It's a randomized placebo-controlled observer-blinded study wherein we have 66 non-pregnant and 556 pregnant women and their infants, and we're conducting that trial in 3 stages. Stage 1 is being evaluated in GBS6 is being evaluated in comparison to placebo in non-pregnant women in South Africa. Stage 2, we're evaluating immunogenicity and safety in pregnant women using various GBS6 doses and formulations compared to placebo. And then for the 3rd stage, we're expanding to the UK and the United States, again, evaluating GBS6 with a selected dose and against placebo. And this study is ongoing. Preliminary data have been submitted for publication, and we look forward to sharing that with the community. Next slide, please. So the next thing that we're thinking about is the path forward to licensure for a GBS vaccine. Given the relatively low incidence of invasive GBS disease in neonates around the world, this prevents conduct of an efficacy study for the following reasons. So if you look at the plot here, we're looking at the number of pregnant women that would be required for an efficacy study with 90% power, 1-to-1 randomization, and a 5% type 1 error. If we were to conduct the trial in the United States with a disease rate of around 0.5 per 1,000 live births, this would require an efficacy study of around 225,000 pregnant women and then their infants. If we were to conduct the trial in South Africa, where the disease rate is significantly higher, this would reduce the trial size, but it would still be a reasonably large trial of around 40,000 mothers and their infants as well. And so this presents various logistical and operational challenges, as well as the amount of time that it would take to conduct such a study, given the low incidence rate of disease. So the field has been exploring whether seroepidemiology studies could be used to determine a surrogate biomarker of protection that could be used to evaluate a GBS vaccine in clinical trials. Next slide, please. And so there is precedent for using a natural history study to define correlates of protection to develop vaccines targeting encapsulated pathogens. One of the first studies was with Haemophilus influenzae, where a natural history study was used to license to develop a correlate of protection to license the vaccine. For pneumococcal vaccines, originally the Prevnar, which was the 7-valent pneumococcal vaccine, was evaluated in a disease endpoint trial, and this led to the development of immunological correlates of protection that could be used to evaluate extended valency Streptococcus pneumoniae vaccines using an antibody threshold that had been defined based on those disease endpoint trials to be associated with protection against disease. And so we're looking to follow a similar path for Group B Streptococcus, wherein we conduct a natural history study to define the antibody levels that are required for protection against disease in infants, and then use those antibody levels to evaluate against the antibody concentrations derived from the phase 3 study in an immunological endpoint trial, followed by a post-licensure marketing commitment, wherein we would evaluate the efficacy of the vaccine against disease. Next slide, please. And so there have been various studies looking at the correlation between antibody level in infants or their mothers, and protection against disease. And these are outlined here. Various studies, all these studies here have defined protective antibody levels that in mothers or infants that are associated with prevention against disease for serotype 1A and serotype 3, which together constitute around 80 to 90% of disease cases. If you hit the next button, please. I've got a next one again. You could just, yes, thank you. Though, as you can see here, these protective levels vary significantly between 0.5 and 6 microgram per mil in maternal or infant sera, and differences between the way these studies were conducted and the assays that were used to define those protective antibody levels prevent an absolute comparison between them, as well as the vaccine that's in development. Vaccines are in development. So a standardized assay to measure IgG concentration could provide a link between seroepidemiology studies and the vaccines in development to establish protective antibody thresholds for each vaccine. And that's really what's being needed by the field. Next slide, please. So we've developed a 6-plex GBS antipolysaccharide IgG assay that's based on the Luminex platform to quantify IgG in serum from infants. So this assay allows for multiplexing. We are able to cross-standardize serotype-specific antibody levels in order to enable direct comparison of the titers across serotypes. And this assay is what's being used to measure the primary endpoint for our clinical studies to assess the immunogenicity of GBS6. And by having a harmonized assay that can be used by the GBS research community, this will enable comparison of results across each different seroepidemiology studies, as well as a link to GBS6, which is in clinical development. And importantly, this assay is being developed by the International GBS Consortium as a standardized assay for quantifying IgG for use in seroepidemiology studies. Next slide, please. So when we think about designing a natural history study to define an immune correlate for anti-GBS CPS, there are various pieces that we need to take into consideration, right, which are outlined here. And just to highlight a few of them, it's important to have the serotype of the GBS isolate that was responsible for infections, so that we can measure serotype-specific antibody levels against that organism. Cord sera, which is a proxy for infant sera at birth, and this is more relevant than using maternal titers because antibody placental transfer can vary depending on various factors that influence the amount of antibody that's transferred from the mum to the infant, such as HIV or malaria. It's important also to have maternal colonization, as this is a key risk factor for infants developing disease, and that enables us to define controls for such studies. And as I mentioned, the validated serological assay is also an essential feature of this process. Finally, the sufficient number of cases is required to enable statistical power for defining protective antibody levels. Next slide, please. So as I mentioned, a global efficacy study would require tens of thousands, at least, of pregnant women, and would be logistically complex and lengthy. And based on the relationship between IgG and risk of disease, this provides a basis for a surrogate endpoint for licensure. And outlined here is our strategy, where we're using a natural history study to define protective antibody levels associated in infants at birth. And our phase 1/2 study in pregnant women, which will measure antibody concentrations elicited by GBS6. We're analyzing both of those studies using the same standardized assay to identify protective antibody levels for infants and compare that to the antibody levels associated with GBS6. We'll use that to define prospective serological endpoints for an immunological endpoint trial, where we would then license the vaccine on the basis of immunogenicity and commit to a post-approval measure effectiveness study, wherein we measure the effectiveness of GBS6 to protect against invasive GBS disease. Just to point out that the unmet medical need for such a vaccine is recognized by key regulatory agencies. GBS6 was granted European Medicines Agency PRIME designation in April 2022 and FDA Breakthrough Therapy designation in August 2022. Next slide, please. So in conclusion, GBS6, as we've shown it to be safe and well tolerated, it's induced robust immune responses in non-pregnant adults. We're using a natural history study to define a serocorrelate that can be linked to vaccine clinical trials to evaluate predicted efficacy. And our phase 1/2 study is ongoing, and that's taking place in the United States, South Africa, and the United Kingdom in pregnant women and their infants. And we're excited about moving forward with this program. Next slide, please. And just since I'm talking here to the young ISSAD group, I just want to give a little perspective about industry and how we work. R&D requires large, diverse teams, and it's dependent on a range of different external groups. Within Pfizer, we have research manufacturing, we have clinical development, we work with lawyers and communications. Really, this is the whole development team involved in getting a vaccine to market. It's also important for us to liaise with academics and investigators, as well as global healthcare organizations, such as ISSAD, and patient support groups to really hear from them and to move these vaccine programs forward. Next slide, please. And a little bit about myself. I have a background in academic research where I focus on molecular microbiology. I gained my MBio in molecular and cellular biology from the University of Bath in the UK, where I focused my undergraduate dissertation on Streptococcus aureus. Staphylococcus aureus, excuse me. My PhD in infection and immunity focused on malaria research at University College London, UK, and I did a postdoc in molecular microbiology and parasitology at Albert Einstein College of Medicine in the US, and I've been at Pfizer for about 6 years now. And my career is really dependent on support from my family and friends, shown on the right, and my team in the lab who are working hard to develop new vaccines, and outlined here and shown here at a team building event. Next slide, please. So just a couple of acknowledgements. Our trials phase 2 study is supported by the Bill and Melinda Gates Foundation and is being conducted in collaboration with the University of the Witwatersrand, Johannesburg, and thanks to the trial participants, the CROs, and the clinical sites that we work with. Thank you very much. So I'll pass over to Dr. Simon Procter at London School of Hygiene and Tropical Medicine.
Thank you, Natalie. So my name's Simon Procter. I'm a research fellow at LSHTM. My work is on a mixture of health economics, infectious disease modeling, and particularly of vaccine preventable diseases. So next slide, please. So today I'm going to be talking about the global impact and cost effectiveness of a maternal GBS vaccine. Next slide, please. So this is actually the result of work which was just published just last night in PLOS Medicine, and I've included a link here if the slides are being shared with people for the paper. And I'm going to start by acknowledging my co-authors who were involved in this work, particularly Professor Joy Lawn and Professor Mark Jitt. And this work really comes off the back of a wider collaboration looking at the global burden of GBS. And this study would not have been possible without a lot of collaborators involved in that wider piece of work. This work was also funded by the Bill and Melinda Gates Foundation, so we thank them for funding. Next slide, please. So what's the motivation for this particular analysis? Well, as I mentioned, we published last year in Lancet Global Health updated estimates of the burden of GBS globally, which shows the substantial burden of disease across all regions. And we've just heard a lot about the development of one of the vaccines that's in clinical trials. My understanding is that the first phase 3 trial of GBS vaccine is currently being planned. And the WHO's meningitis roadmap has identified licensure of a GBS vaccine as a priority with a target date of 2026. So why are we undertaking this economic evaluation? Well, partly to inform the significant investments that will be required to bring a GBS vaccine to market, but also to understand how the cost effectiveness of a GBS vaccine might vary between countries and regions in order to enable work to be done to put in place funding to ensure equitable access to vaccines when they do come to market. And this study is part of a wider program of work which has informed the World Health Organization's GBS full value of vaccines assessment, which is available from the WHO website. Next slide, please. So I'm just going to give a quick overview of the model. We don't really have time today to go into lots of detail, but we use a decision tree model that's designed to reflect the natural history of GBS infection, which includes the different important clinical outcomes of GBS. So we have in this model stillbirth, preterm birth outcomes, early onset GBS infection, late onset GBS infection, and then we have long-term sequelae in terms of different levels of neurodevelopmental impairment following either sepsis or meningitis in survivors. The risks of these outcomes in our model are parameterized at the country level based on the results from the global burden analysis that I mentioned earlier. So we model a cohort of 140 million pregnant women across 183 countries and using demographics for the year 2020. And since the vaccines are currently in development, we base the vaccine impact in the model on the preferred product characteristics that have been developed by WHO. And so we're assuming here in our base case a one-dose vaccine that's 80% efficacious against early onset GBS, late onset GBS, and GBS-related stillbirth. We then assume coverage in this model based on the percentage of women who attend 4 antenatal care visits, assuming that vaccine delivery would be through existing antenatal care platforms. And then the principal outcome of this model, we calculate something called the net monetary benefit of vaccination. And that net monetary benefit essentially takes the both cost savings from averted disease and healthcare, together with the benefits in terms of quality adjusted life years gained, and then monetizes them to turn them into a dollar value. So if that net monetary benefit is positive, then that reflects an overall economic gain and a cost-effective vaccine. And it's important to point out that the burden estimates which we use in this model reflect the current practice in different countries in terms of IAP. And so what we're evaluating here is the additional value of a GBS vaccine on top of whatever the existing IAP policy is in a given country. Next slide, please. So we include costs for the vaccine program. So we include the purchase price of the vaccines are shown in the table here. So we assume different prices based on the World Bank income region that a country's in. And these prices for our base case analysis are based on benchmarking against other vaccines. And in addition to that, we include delivery costs, based on a previous systematic review of the cost of delivering maternal immunization programs. And then we use a regression model to extrapolate those results across countries. Next slide, please. We also include the GBS healthcare costs. So we include, so we're in this analysis including only costs from a healthcare payer perspective. So we're excluding other important costs of GBS disease to families and productivity losses and other costs to wider society. So there are few studies which have looked directly at GBS healthcare costs. So we base the acute costs in this study on a systematic review looking at the acute costs of sepsis and meningitis in neonates and young infants. And then we also model the annual recurring costs amongst those individuals who develop long-term impairment as a proportion of the cost of the acute episode. Next slide, please. And then to estimate the QALY loss, we calculate firstly, we calculate the QALY loss associated with stillbirths and deaths using the expected average life expectancy in each country. And then we include also the QALY loss associated with the acute meningitis or sepsis episode, as well as an annual QALY loss for survivors with neurodevelopmental impairment. And we base the, so again, there are very few studies that have estimated the health state utility values for these long-term outcomes. So we use the values from a UK study which looked at different levels of neurodevelopmental impairment in young children and teenagers in the United Kingdom. We additionally include a QALY loss associated with preterm birth, which show based on a systematic review that shows that on average all preterm births have consistently lower quality of life scores compared to term births. Next slide, please. So in order to convert the quality adjusted life year outcomes into dollar values, we use a variety of alternative normative assumptions. So these are different assumptions that might be made by national or international decision-making bodies when they are undertaking economic evaluations. So we define a least favorable set of assumptions and a most favorable set of assumptions. So for the least favorable assumptions, we assume discount rates for costs and benefits of 3% and assume that these are both the same. Whereas for the most favorable assumptions, we assume that the benefits are not discounted, but that the costs are discounted. There are differences in the positions taken by decision makers on whether or not QALY loss due to stillbirths are valued or not in health technology assessments. So we include those alternative assumptions. And there are also differences in the cost effectiveness threshold that are sometimes recommended for deciding on whether or not an intervention is cost effective. So we use both empirical estimates and also a 1 times GDP per capita estimate that has been commonly used in the past. Next slide, please. So looking at our headline findings, we find a global vaccine program in 2020 could cost $1.7 billion, but save around $300 million in acute healthcare costs and $85 million per year in long-term healthcare costs. Globally, a high coverage vaccine program could avert an estimated 127,000 early onset cases of GBS, 87,000 late onset cases of GBS, and around 30,000 infant deaths and around 18,000 cases of moderate or severe neurodevelopmental impairment. Additionally, if a vaccine is effective against GBS stillbirth, we estimate that this could, a vaccination program could prevent around 23,000 stillbirths. There's also, in a sensitivity analysis, there's also the possibility that a GBS vaccine may prove effective against GBS-associated prematurity. And if that's the case, we estimated around 185,000 preterm births might be avoided, although this is subject to very wide uncertainty. Next slide, please. So we estimated that the global net monetary benefit ranged from 1.1 billion pounds to dollars to $17 billion, depending upon the normative assumptions made. So under the least favorable assumptions, if the value of stillbirth QALYs are added in, this almost doubled the net monetary benefit. Next slide, please. So we also estimated the net monetary benefit at the regional level. And under the most favorable normative assumptions, we found that NMB was positive in all regions, but under least favorable assumptions, estimated NMB was negative in some regions. And that reflects the distribution, or that, well, both the uncertainty in some of the parameters, but also heterogeneity between countries within regions. Next slide, please. So this slide is quite busy, but the panel on the left shows each dot on the panel on the left is a country result, and on the y-axis it's showing the probability that the vaccination would be cost effective in that country. And the countries are ordered here by GDP per capita, so wealthier countries are towards the right-hand side. And what we see is, as with the regional analysis, under the most favorable assumptions, then we find vaccination will be cost effective in almost every, essentially in every country. But under the least favorable assumptions, the vaccine was less likely to be cost effective for poorer countries within a given region, suggesting that the vaccine price will need to be carefully tiered to ensure equitable access across countries. On the right-hand side, this figure is showing the threshold vaccine price. So this is the price at which a vaccine would become cost effective. And this is showing the distribution of vaccine prices across each World Bank across countries within each World Bank income group. And what you can see from this is for almost all countries, even under the least favorable assumptions, there is a vaccine price at which the vaccine would be cost effective, suggesting, but for some of the countries, this suggests that the price of the vaccine would need to be subsidized below the price that we've assumed in our base case analysis in order for that to be the case. Next slide, please. So our results are subject to a number of limitations. So there's wide uncertainty in some of the model inputs and therefore in our estimates based on the uncertainty in the epidemiological parameters which underpin the burden estimates. And there's also wide uncertainty around some of the cost inputs because there are not many studies that have directly estimated the healthcare costs for GBS. It should be noted that we've not estimated the wider impact of GBS, such as cost to families and caregivers and productivity losses, which would increase the value of a GBS vaccine from a societal perspective. It's also important to note that we've not considered scenarios where a GBS vaccine replaces existing IAP practice. And if that were to be the case that could happen, then potentially that would allow additional cost savings in these settings, which would increase the value and cost effectiveness of GBS vaccines in those settings. And finally, there is obviously currently uncertainty about the characteristics of GBS vaccines that may be licensed in the future. Next slide, please. So in conclusion, globally, a GBS maternal immunization program is likely to be cost effective and avert a substantial burden of death and disability in children. At a regional and country level, cost effectiveness is sensitive to vaccine price and to different choices policymakers may use to value the benefits in improved health. Our findings therefore highlight the need both for carefully tiered vaccine pricing to ensure equitable access across countries, and also for local assessment of cost effectiveness as GBS vaccines move towards licensure. And finally, there is a need for more evidence on the impact of GBS on several outcomes, notably GBS stillbirths, preterm births, and maternal morbidity, as well as the wider societal costs of long-term GBS-related disability. Thank you for your attention.
Thank you, Dr. Simon and Dr. Natalie, for these two excellent and very informative presentations. So thank you for accepting our invitation and for being here today. We have some questions in the question and answer box that I will read for you. We have questions for both of our speakers, and I encourage all of you to send questions and comments in the question and answer box if you wish. So the first questions are from Dr. Natalie. We have 3 questions from Dr. Oliver Plumb. I think I will read one of them at a time, Natalie, for you can answer separately. So the first question is, what lessons have been learned about maternal vaccine rollout from the COVID-19 vaccines, and how can those be applied to a GBS vaccine?
Yeah, that's a great question. I think the first piece is that lessons from that, as well as the long history of maternal immunization, is that maternal vaccines are safe and effective at preventing disease in infants, as well as impacting the mother, which would be, another benefit of a GBS vaccine that we're developing. I think that another piece is that we really have the technology and the scientific expertise to develop maternal vaccines. But the next part, and this speaks to really what Simon was talking about with cost effectiveness and rollout in low and middle income countries, Pfizer has a broader commitment to work with the global health community to accelerate global access to vaccines, especially in the world's poorest countries, and to make them affordable and sustainable. And so that's really something that's going to have to be, especially because of the burden of GBS disease in low and middle income countries, that will have to be really thought through. And that's why we're also working with the Gates Foundation for developing this vaccine.
Yeah, thanks. Next question is, what's Pfizer's best estimate of when GBS6 will be available and licensed for use?
Yes, I'm sure everyone would love to know that.
Yes.
I can't give an estimate, unfortunately, but the next stage is to move into phase 3. That we're really excited about the prospect of doing that, and that will take 1 to 2 years at least, and then licensure would follow, and that will be applied for in various countries and groups of countries around the world. So a few more years, but hopefully it won't take too long.
Yeah, hopefully. The last question from Dr. Plumb is, would high-income countries like the UK, which are still using risk factor prevention strategies, be key targets for GBS vaccine introduction, given their higher burden of GBS disease?
Right. So I think that's something that will have to be carefully considered. The UK, for example, and the US, both high-income countries, are included in the phase 2 study, and it's for that the UK for that reason, as well as others, that there's a higher burden of GBS disease. So it will be important to assess predicted efficacy and safety as well in those countries. But yes, good point.
Next question is from Dr. Hamadevi Hagunandan. And the question is OPK-A, I believe it's OPK-A, not considered for clinical endpoint?
So that's something that we're working through, but there are some limitations to using OPAs in these types of samples. They have limited sensitivity, and that's been shown by a couple of studies from GlaxoSmithKline, looking at the correlation between IgG and OPA. You don't see OPA activity until there's reasonably high antibody levels. And so that would make it very difficult to define a protective level of OPA associated with disease risk. And there have yet to be any studies doing that. There have been a number of studies over the past 10 plus years looking at the association between antipolysaccharide IgG concentration and risk of disease. And that's very well established, but for OPA, it isn't. So I think it'll be important to show that you know, there is OPA activity associated with antipolysaccharide IgG concentration, but there are limitations. Another is that comparing across serotypes can't be done with OPAs, and so that presents another challenge especially when you're looking at serotypes that have very low numbers of cases, in order to be able to define the relationship between risk of disease and IgG. So it's something we're thinking through, but focusing on IgG.
Okay, thanks, Natalie. Next question is for Dr. Simon from Jane Plumb. So Jane said, fascinating presentation from Simon Procter. His paper may well answer this question, though I've not read it yet. So the question is, what would the impact be on cost effectiveness if multi-doses of the vaccine needed to be given? Would it still be cost effective if 2 or 3 doses were needed?
Thank you for that question. It's a good question. It's quite a complicated question. So in the paper, we did look at two-dose vaccination, and if you obviously if you assume that the price per dose remains the same, then a two-dose vaccine would essentially be doubling the cost of the vaccine program. And that obviously has a big effect on cost effectiveness in those countries where cost effectiveness is quite marginal. So that includes countries where the burden is lowest. Now, I think what complicates things a bit is that some of those countries are will be countries with effective IAP. So probably what starts to come into play is this question as to whether or not a GBS vaccine can replace the need for IAP screening and release some of those costs, or obviously if the vaccine can become available at a lower price point, then it can still be cost effective. But this picture really it depends a lot on interacting factors in the at the country level. I think the other thing that could have a really big impact is the question of whether or not the vaccine is effective against GBS stillbirths and GBS prematurity. So most of the impact that we modeled on GBS on GBS stillbirths and GBS prematurity is actually in higher income countries where the burden of early onset GBS is lower. So those factors would also come into play. So yeah, so it depends.
Okay.
It's probably actually worth, sorry, one final point related to this is I think there's an interesting question of if you have a multi-dose, I mean, vaccine, about when those doses would need to be given. So I think if you had 2 doses, there's a question of can you give those 2 doses during one pregnancy? And in the analysis we've done, what we've looked at is the impact of one vaccine dose on one pregnancy. But if you were looking at something where you maybe gave a priming dose during adolescence and then followed by a booster dose in pregnancy, then that would look, that would require a different analysis.
Yeah, thanks, Simon. Next question is for Natalie from Dr. Edmund Badley. So the current challenge with PCVs is serotype replacement. Are we not likely to face a similar challenge with the GBS6 since there are 10 serotypes?
Yeah, thank you for that question. It is a good question. For Streptococcus pneumoniae, there are over 100 serotypes that have been identified. For GBS6, there are only 10, and only 6 of them are associated with the majority of disease. The others, there are very few cases, and so it's less likely because there are less of them. But it is a possibility. I think the other piece to keep in mind is the route of transmission and how colonization is occurring with pneumonia versus GBS. But this is something that would have to be evaluated in post-licensure effectiveness studies to confirm whether there is or isn't an effect.
Okay. Next question is from Stephanie Long from the UK Health Security Agency. Stephanie wrote a comment and a question, so I'll read first the comment because it's related to another question. So she said, just a comment on the OPK sensitivity. She said that their assay at the UK HSA detects to 0.001 micrograms per milliliters dependent on serotypes. So just a comment she made regarding the OPK question. And she wrote another question, which is, will the HIV status of the SA cohort from the phase 1/2 study need to be taken into consideration? Also, whether samples are from infected or exposure.
Yeah, so to address the first point, that the HIV status, we're looking into that and the impact that has. Obviously, HIV affects transplacental transfer, and so that is something that needs to be considered. Importantly, the South Africa cohort and the phase 1/2 study. The South Africa participants are from the same sites, and so there is that comparability between the two populations, and the study was designed in that way. Whether the samples are from infected or exposure, so it's a case control study. So we have infants that develop disease and those that were born to mothers colonized with GBS that did not develop disease. And we're looking at the difference in antibody concentration between the cases and the controls. And the comment regarding the assay is helpful. I can't comment on comparing the 2 different assays or assays within the field. Yeah, thank you.
Next question is for Simon from Dr. Anna Seali. How sensitive are the estimates to ANC4 coverage?
So obviously the total impact and the value of the economic gain is sensitive to the coverage, but whether or not the vaccine is cost effective in a given setting is not very in our model is not sensitive to that coverage. I guess there is a question about if you go from low coverage, low ANC coverage setting or a high ANC coverage setting, how that actually influences the cost of delivery in those settings. But that's something that we don't have detailed estimates for, but I can imagine that they could have some impact on whether it was sort of cheaper or more expensive to actually set up a program to deliver the vaccine in a given setting. But the overall cost effectiveness result should not be very sensitive to the coverage level.
Thanks, Simon. We have more 2 or 3 questions, but unfortunately we need to wrap up and finish our webinar. I'll make sure that I forward the questions to the speakers and then they can reply you. So thank you again, Dr. Simon, Dr. Natalie, for being here today and for your great and informative presentations. So now moving to Carol.
Hi, everyone. First, I would like to thank you also, the speakers, Dr. Simon, Dr. Procter, for the excellent speeches on subjects of high relevance to us. And here I would like to invite everyone to like and follow our social medias. Our next seminar will be in June, and everyone is invited. And you have also another invite to the event, ISSAD event, that will take place on October in Rio de Janeiro. It's a pleasure to have you here to participate, okay? So thank you and to the next.
Thank you, everyone.
Thank you.
Have a good day.
Thank you, everyone. Bye.
Thank you.
Bye-bye.